Okay, hello. So we're here again, chapter 9, but this time we're going to talk a little bit more about pH and then, of course, drug interaction with the body a little bit, at least an initial exposure to that with respect to the pH and whether the active ingredient, for instance, if it's in acid form, would be better able to penetrate into the cell or into the tissue relative to the conjugate base form. So let's get cracking on this. We have here introduced pH, and it kind of dovetails in with the constant of water, with that equilibrium constant for water, which we just discussed at the tail end of the other seminar, part 1 in this chapter. So let's continue on then. We have water at pH equals 7, that would indicate a neutral condition where the acid and its conjugate base would both be present and in equal amounts. So here's the expression again. We have the equilibrium constant for water, which the hydronium ion and the hydroxyl ion are in equal concentration. It's a 1-to-1 ratio, and they're well balanced. 1 times 10 to the negative 14, and this is at like 20 or 25 degrees Celsius, will correspond to that. Now, if you had them both essentially being in equal concentration, then let's just assume then that the hydroxyl group, which is the conjugate base, actually would be essentially the same as the hydronium. So we're just going to assume that we have 2 hydronium ion concentrations. So let's just square those, and that will equal 1 times 10 to the negative 14. Now, if we were to take the square root of that, because we're interested just in the H3O, the plus ion, the hydronium ion concentration, then that's what that would be. 1 times 10 to the negative 14 raised to the 1/2, which is 1 times 10 to the negative 7. If we can follow our rules of exponents, where you just simply divide 14 by 2 to get 7 if it's in exponential form like that. And now we have the concentration being equal to 1 times 10 to the negative 7. Now, before we had introduced the term with the pKa and the Ka, where the pK was the negative log of the Ka. Here we're going to do the same thing, but we're going to substitute H for Ka, okay? So instead of the dissociation constant, Ka, we're just going to use H right here. What does that mean? That's the concentration of the hydronium, which we had just found here above in this other expression. So pH then equals negative log of the hydronium ion concentration, which we already have calculated to be 1 times 10 to the negative 7. And this is only at neutral conditions, again, at pH equals 7. And in doing so, the hydronium essentially becomes that proton, that hydrogen ion, which we have sometimes said that is used interchangeably. To be very pedantic about it, be very specific about it and original, then it's going to be the hydronium ion, but that's not how things work, because that's a lot to say and write. So we use H plus for the shorthand notation of that. So you have negative times net log of 1 times 10 to the negative 7. Anything to the negative power, if you're taking the log of it, it's going to be that negative power itself, especially if it's just 1 times 10 to the negative 7. I mean, this is just easy math here. Otherwise, you'd have to also include like the log form or whatever that, non-value of 1 would be, like if it was 2 times 10 to the negative 7 or 3 times or 3.4 or 4.6 or whatever it would be, then we have to factor that in too. But in this particular case, it's simple. It's just 1. So log of 1 is 0. And the way that the exponent rules would work is that you would add them. So log of 1 is 0 plus the log of 10 to the negative 7, which is just negative 7. 0 plus negative 7 is negative 7. And that's why we have that here. Anyway, you guys can refresh your mind if you need to with respect to logarithms. But this is what you end up getting. You have the pH then is equal to positive 7. Okay, 2 negatives make a positive. I like that math. Another way to look at this is that if you have the hydronium concentration or H plus concentration known, then you can actually set that to be equal to 10 to the negative pH. So we just took the inverse of that, like the inverse power of that, if you are interested in knowing what that would be. So in this particular case, if the pH was 4, it would give rise to a different number, right? 10 to the negative 4 would be the concentration of the hydronium ion, or 10 to the negative 12 if it was a basic form, which actually, would make actually pretty good sense, because you would expect there to be more acid nature to the hydronium concentration. It'd be more concentrated under a very low pH, which would indicate acids are present, versus a higher pH, which would indicate a shift towards bases. So for the table 9.7, this is in your book, but it's also shown here. If pH is greater than 7, then it's basic, okay? The hydronium ion concentration will then be less than 10 to the negative 7. If it's acidic, the pH is going to be less than 7, and it's going to be greater than 1 times 10 to the negative 7. It's going to be neutral. It's going to be 7 exactly. So we're setting water to be the de facto reference point here. So everything is going to be with respect to water when we're talking about pH. If you're going to use a different pH scale, then you're going to have based on a different reference, then it is going to be different. Okay, so if you're not using water as the reference, then there's going to be some shifting in values. But for all intents and purposes, and for our lives, we're only ever going to use water as that reference point. So you have pH plus POH equals pKW, which is equal to 14. So that is how that works. You can kind of massage all those different logarithms as needed in order to try to figure out what that means. So here would be a scale going from 10 to the 0, actually 10 to the first power. This would be in particular concentrated hydrochloric acid. Remember how I said it was like, I think up to like 48% or so concentration. Muriatic acid's definitely in the like low 30s. I use 7% hydrochloric acid in that one neutralization experiment with hydrochloric acid. So you have acidic environments, which are generally going to be greater than 10 to the negative 7 in terms of actual numbers, like 10 to the negative 2, right, is larger than 10 to the negative 7. And then smaller numbers are going to refer to the basic composition. And then on the right-hand side of this pH kind of indicator, you have a bunch of different materials, compounds, and just solutions and whatnot. This actually is going to dovetail nicely with the lab that you guys will be doing, where you have to explore the effective pH in response to different components there. So pH is generally determined using a meter. You could do that very, very sensitively. You know, it's much more precise than using a pH strip, but you can also use pH strips. If you're measuring, for instance, pH of a pool, you would probably want to use a pH strip. There are some portable meters that you can also use, which are really effective. The only problem with these meters is that, and I don't want to say a problem, it's just a condition, you always have to reference it. You have to recalibrate it with respect to known solutions of known pH. And if you do that, then it'll all be good. So it's just one of those examples. pH paper is very popular because you don't have to do any referencing or calibration. To take the strip of paper, you dip it into the solution, and then you see what the color is going to be, and that's what your pH is probably going to be. So that's just part of it. pH is also indicating phenolphthalein. Okay, phenolphthalein has been produced and continues to be produced by certain fungus actually, and it's just part of the pigmentation of it. It just ends up being a pretty good pH detector. Anyway, table 9.9 actually here, before we go on, shows you the relationship between the relative amounts of acid and conjugate base, pH and pKa. So this is really important. So, and we're going to be revisiting this several times over the next several slides, but this is going to be really important because this is going to allow us to better understand what form of a compound is existing in solution, whether is it the acid or whether it's the conjugate base. And so all this stuff is going to have a big impact on what we're doing. So you have pH is less than the pKa, then guess what? The acid form predominates, okay? If the pH is equal to the pKa, well, acid and the conjugate base of whatever we're talking about is like, let's just say citric acid, that's going to also be the same, as long as the pH and the pKa are right around the same amount. And if the pH is greater than the pKa, then the conjugate base is going to form. So we're going to get into that, and I'm going to show you examples of that. But first, I want to point you to another YouTube video that I did demonstrating the sensitivity of food spice to pH. So I just talked about like fungus and other types of like living plant matter can actually be used for pH indications, and it's just because some of their molecular makeup is made with compounds that are just sensitive to pH environments. It's probably not surprising to you guys because pH is always going to involve loss or shifting of protons, basically. And so because a lot of organic components have functional groups that involve protons, like, I don't know, alcohol groups, carboxylic acids, you might even have some esters, you might have some aromatic character, all these different things, then it's probably not surprising that some of the things that are found in nature could actually be useful for pH indicators. Here is one of the examples where it is. The spice turmeric, okay? The key ingredient in turmeric, okay, this is a very like orangish brownish kind of spice. It's not very soluble in water. I mean, it can be just slightly, but it's usually more soluble in more of a non-polar based environment, or environments that favor maybe a dipole-dipole kind of interaction, something like maybe an ether or a long chain alcohol or something like that. Even a short chain alcohol in this particular case might actually work well for its dissolution. So the key ingredient in turmeric that gives rise to any kind of action is the curcumin. And this is the molecular weight. It's a C21H20O6. It's a pretty big molecule. You can see some of these have been given here. And don't worry about this diketone or the ketoenol form specifically, but know that the only difference in this entire large molecule, and it's kind of interesting, is right in the middle of the molecule, you have two carbonyls for the ketone, which they consider a diketone. So that's probably not surprising to you guys, because you already know a bit about that carbonyl functional group being present in ketones. But the really interesting part here is the enol. We never really talked about this because this is a little bit more advanced, but the enol involves an alcohol group, okay? It involves an alcohol group like the OH, along with a very nearby, an adjacent double bond, an alkene environment and an alcohol group. And what happens is that alkene environment can open up and respond to, well, in this particular case, electron transfer or, pH environments. And that can arise and develop even if you start with a carbonyl like you did before in the diketone, and that conjugation can actually shift down. So one of those electron pairs can actually just switch down into the carbon-carbon bond, and in fact then creating a double bond, but then you still gotta conserve all your environments, and then hydrogen is actually very prone to interacting with that if it's only a single bond between carbon and oxygen. So it just happens to be like that. That's just the nature of this functional group called an enol. It's just an interesting aspect. So I mentioned that. So curcumin is sensitive to pH, ketone versus alcohol functional groups, and it generally happens at or around pH of 8. So what do we mean by that? Again, you have to watch the video because you'll be able to see the different colors that have been created depending on what environment that this turmeric was exposed to. So you'll get a sunlight yellow if it's added to some citrus acids, and it stays more or less this kind of neutralish, slightly, basic pH at the brownish orange, color if you were just to add it into, like, I don't know, tap water or something, tap water, reverse osmosis water maybe, although reverse, RO and distilled water tend to be, I don't know, they may not be as basic per se. They might just be slightly more acidic or slightly more basic, I guess, depending on the system and what's used to be able to get that, but it does vary. And then of course, if it does go basic, it actually turns into this beautiful crimson and wine color, or even like a kind of like a sienna color if it's not too too basic. So it's pretty interesting. So watch the video, you'll be able to get an indicator there that turmeric, a common spice ingredient, can function as a pH indicator. It's pretty neat. And then here's another one that I think is pretty interesting, and it's the bromothymol blue, okay? And there's another video, and it has a different link. You can go to it and check it out. But this sensitivity of bromothymol blue is even wider with respect to the pH range to, whatever solutions that you're trying to understand. In general, it's going to have like an aquamarine kind of appearance to it. It's a pretty big molecule, relatively speaking, to what we have been accustomed to. So the molecular weight's 624, so it's almost double what it was for curcumin, okay? And the keys here is that it has two bromines and it has a sulfur also attached. Actually, bromothymol blue is just one of the dyes in the class of these sulfone phthalates, right? that are actually, again, existing in nature. This is the general structure. You have 3 aromatic regions, and you have different functional groups attached at different points along those aromatic regions. With respect to the bromothymol blue, the R1 position has an isopropane attached to the R2 is a bromine, and then R3 corresponds to methyl groups. But there's a lot of different ones. There's at least 10 different forms of these types and of these class of dyes actually that can be used for pH indicators. And they are, and they're used in biology, they're used in biochemistry. I mean, a lot of different ways. And I wanted to use one here to give you guys an example of how this is pretty robust in terms of being sensitive to pH. It is a weak acid, and it is sensitive to pH with respect at acidic environments. It becomes protonated. If it's neutral, just essentially is like an as-is kind of thing. The pKa is around 7, which is right around that neutral aspect of pH, right? Isn't pH of 7, isn't that neutral? Well, the pKa value of bromothymol blue is also 7. So you're going to have equal amounts of the acid and conjugate base, or maybe just slightly shifted more toward the conjugate base, or maybe shifted a little bit more to the acid, depending on how close to pH of 7 one actually is. And then if you have a very basic environment, it's basically deprotonated. Well, what does that mean? So you have several different examples of what that is. I left the basic pH blue over here on purpose empty. We'll get to that in a minute. You have the neutral pH, which is kind of like a greenish yellow, and we have to look at this functional group. Guys, this is something that you haven't been exposed to yet, but this is called a benzoxethiol, okay? And it's an SO3, but the oxygen is actually attached to this carbon, this saturated carbon, which is also attached to 1, 2, 3 aromatic species. So that's where kind of it starts, and you can see that up here in the upper right-hand cartoon of that. You can see almost, maybe even a little bit clearer depending on your view. And again, in the slide deck, you can expand this if you want. But you'll see you have these 2 S oxygen, right, double bonded structures here. The 2 of them almost look like they're legs or whatever. They form like this carrot kind of shape, but then you have these arms coming off that sulfur, and one of them is directly attached to that aromatic, and then the other one is just attached to this oxygen, which is further attached to a carbon. So under different conditions, especially under acidic conditions, that carbon-oxygen bond breaks, okay? And then you're left with a decidedly negative sulfate head group there, okay? And that is perfectly reasonable. You have, essentially a protonated environment, because that bonding environment has definitely changed with the loss of that oxygen there connected to the 1 carbon, right? So that's pretty interesting. That pales in comparison to under basic conditions, where although you still have the SO3 moiety still present, and by the way, in the basic condition, you have 2 different structures that form. And by the way, these are essentially enantiomers of one another. They're left and right. It's like left hand and right hand. And you can't overlap them because they have different orientations. So these are a certain type of isomer. But look, both of these are equally predicted and observed, and I give you references here, that kind of show that. But the interesting part about this, because you're probably wondering, well, where's the protonation coming from and where's the deprotonation coming from? Well, just look at the oxygens nearby, and I highlighted those in red, okay? So without the hydrogen present, remember with weak acids or even strong acids, the hydrogen is going to be splitting, right? Well, where's the hydrogen coming from? It's coming from this hydroxyl group that's attached to one of the aromatics, okay? And without that aromatic present, without the, in a basic environment, that hydrogen is lost. And in doing so, you actually get essentially the conjugate base of this bromothymol blue. So that's where that's coming from. So take a look at the video, because in the video, you're going to be able to see some really clear differences in color with respect to acid, neutral, and basic environments. And again, I'm going to, refresh this with the pH is greater than pKa. The conjugate base form presents itself. So again, here with the bromothymol blue, if the pKa is also 7, as it is with 7, then you know that you're going to have a conjugate base form dominating. And that would be then in this pH area here with like pH of 11 or 12. For instance, I use sodium carbonate again, which has a pH of around 11, 11.5, and that is what actually helps give rise to this blue color then. pH of 11 is definitely greater than 7, so you're going to get this conjugate base that is going to dominate. With respect to the acid form, I use formic acid, the same stuff that's delivered in an ant bite, okay? And that has a lower pH, less than 3, and the pH is less than the pK, so the acid form is going to dominate, okay? So it's a pretty interesting observational kind of demo. I would highly recommend you view that and get some information on it. So now let's talk a little bit more about pKa and pH. So at physiological pH, which we take around 7.4, just, you know, so is it perfectly neutral? No, but it's also not really basic either. I mean, things that are going to be pH 8 and higher are going to have a decidedly basic character. I would consider 7.4 still to be right around neutral, okay? But which version of acetylsalicylic acid exists? So here's acetylsalicylic acid, and this is also known as aspirin, okay? And you see the carboxylic acid functional group at the very top of this molecule. You also see the ester part, right, with on the right with the 2 oxygens, right, one of the carbonyl shapes. So you have the carboxylic acid, you also have an ester, and you also have an aromatic or phenol group. So at pKa of 3.5, that means that you know, you have the dissociation constant that will occur at 3.5. You got to relate that to the pH, and then that's going to dictate which hydrogen's coming off. So at the pKa value, okay, where do you think the hydrogens are going to come from? They're not going to come from the aromatic region. They're also not going to come from the saturated, you know, areas where you have, you know, carbon-hydrogen bonds from the methyl. It's going to come from that hydroxyl group, the OH. So the conjugate base of acetylsalicylic acid is going to be all this entire molecule, but without that hydrogen, right? So what do you call it? Acetylsalicylic. Okay, and so this is what you have. The hydrogen pops off. This is the conjugate base. So this is the question that you must ask. If the pH then is below or higher than the pKa, which form will then dominate? Well, if the pH is below pKa, then the acid form will dominate. So is our body at a pH less than pKa? No, it's actually higher. We're at 7.4. So because we're at 7.4, our physiology is high enough to encourage dissociation of the acetylsalicylic acid, and so this conjugate base form dominates, okay, in our blood. Now you might say, well, in the environment of the stomach, it might have the pH is less than 3.5, and that is absolutely true. So whenever we take this, it's still very acidic, and the environment does not promote, in our stomach anyway, the dissolution of the acetylsalicylic acid, meaning that we're still going to maintain that carboxylic acid functional group there. But as it progresses from the stomach to the small intestine to the large intestine, and then ultimately into our blood, where the pH continually increases, well, guess what? That hydrogen will pop off, and it will be able to circulate in our body in the conjugate base, and therefore the quote-unquote active form. See, I think it's important. All this discussion about the stomachs and the intestines from the previous webinars definitely help our understanding of what might be going on. So let's take a look at amino acids. These are generally regarded as biologically weak acids. So here's alanine. It has a protonated amine, and it also has a carboxylic group here. The pKa of this carboxylic group right here is 2.3. And the pKa of this protonated amine, again, all in the same amino acid, is 9.7. So alanine exists as a Zwitter ion, we call it, meaning it has both negative and positive character at physiological pH. How is that possible? Well, isn't the pH of 7.4 greater than pKa? Yes. If it is, then the conjugate base forms, which is what this is, the carboxylic acid, or the carboxylate, as shown here with the negative on the oxygen. And what about over here with the protonated amine? Is the pH 7.4 is a higher or lower than the pKa of the protonated amine? right? It's lower. So this is going to dominate. So it's just, it's very helpful to try to make sure that you keep the pKa and the pH levels in mind as you're trying to better understand, what form of the molecule is going to exist at, at a given pH, I guess. They call it the isoelectric point, where the Zwitter ion, the positive and negative, are both present and no net charge results, okay? So let's take a look. If you had the alanine ion just like this, all right, and notice that you have the pH is more acidic than 6, okay? So we haven't lost that proton yet. Let's just only focus on just having the the alanine ion with that protonated amine, okay? It has a net charge of +1. How is that possible? The pH is less than 6, okay? I know that you might be saying, well, look, I'm trying to understand the pKa value of this carboxylic acid group right here, and you say that it's 2.3. Well, no, we're actually talking about the entire molecule, okay? We're not just talking about just the carboxylic acid component here, okay? So hang with me here for a minute. The isoelectric point, actually, of alanine in general is at a pH of 6, okay? Just like how the neutralization of water is at pH of 7, here for alanine, that isoelectric point where you have no net charge, that's going to be at the pH of 6. When I say no net charge, that doesn't mean that there aren't charges. It just means that the positive is balanced equally with the negative, okay? So if base is added because you started with a very acidic environment, and now you're gonna try to get to the isoelectric points, and you're gonna add some base, whatever that is. It could be carbonate, right? Especially if it's in your blood, it's gonna be carbonate. If it's outside the blood, you might add, I don't know, some ammonia, maybe some sodium hydroxide. You have to be careful though how much, just use diluted amounts of that base, get it to this point where you're at pH of 6, which happens to overlap that of the isoelectric point, and then you're going to get both charges existing. If you're going to go back the other way and you want more of an acidic charge, well, just add some acid. So dilute hydrochloric acid, for instance, or maybe you want to add some citric acid or another carboxylic acid, maybe acetic acid or lactic acid or something like that. That's fine too. Now, under conditions where you have pH is greater than 6, what do you think happens? Well, you continue to add some base, right? But then you're also going to get well above that point where the carboxylic acid form will now start to dominate, okay? And so here the alanine ion is, the pH is more basic than 6, and so you have an overall net charge. So that's what you're looking at right there. So hopefully that makes some good sense. But again, you can go back each way. Just keep track of where where you're starting from and what you're doing to add in the protons or the hydroxyl groups. For instance, if you're already at, let's just say you're at pH of 7.4, right? So if you're at a pH of 7.4, you already know that your carboxylic acid is going to be the dominant species anyway, right? Because it's greater than that 2.3. And it's also greater than the 6, which is the isoelectric point. And also at that stage, because you have an NH2, there is no net charge, okay? So 7.4 is less than 9.7, so you're gonna have a non-charged environment over here with this amine. Now, if you add some H plus by virtue of maybe you added a little bit more citric acid, all of a sudden you start making things a little bit more interesting, okay? Where you are going to start having that protonated amine develop, especially near the pH of 6. If you keep on adding it, then you're going to get to the point where the carboxylate form will not exist, but you're only going to get the carboxylic acid form of this. So it's just something to be familiar with, okay? So pretty neat. I think that this is really helpful. Again, here the pH is greater than pKa, conjugate base is favored. The other one, the pH is less than the pKa, so the protonated amine is favored, and we're just going from there. And look at the pKa. We're trying to talk about that with respect to the protonated amine, okay, environment. So hopefully that will help you. Now, let's take a look at tissue pH and drug delivery, and we're going to use the example of lidocaine, okay? How do you deliver a drug which is usually polar across a non-polar membrane? So this is where it is becoming really important to apply everything that we've been learning about polarity, some of these molecules and their functional groups, and now we're actually trying to use, you know, a real life example here. You use compounds that are reasonably soluble, yet have enough nonpolar character to cross the membrane. Remember, phospholipid bilayer, you have a nonpolar central core here that has all those tails, and on the outside and inside of the cells, you have a polar environment. If you have lidocaine, which is a local anesthetic in the protonated form, okay, RNH+, you see how there is a protonated amine structure right here on the end. You can notice that the rest of the molecule is very nonpolar, isn't it? You have, I mean, yeah, you might be able to get maybe some hydroxyl bonding around this nitrogen that has the hydrogen with it in the middle. But overall, this larger aromatic structure kind of negates that, and as well as the carbonyl. The carbonyl also kind of frustrates that. But as soon as you put in that charge, all of a sudden things become a little bit shifted more towards the ion dipole type of interaction, because obviously there's going to be a very moist, i.e. watery environment, and so you can expect there to be ion dipole interactions. So we got to take a look and see, well, you know, what kind of form are we talking about? And whenever we use the term R here, that just means the rest of the molecule. We're talking about this protonated amine, right? NH+, and then R is the active, or R is the rest of the molecule. If we're talking about the active form, that's how it's written. The diffusible form, though, is going to be the rest of that molecule with the nitrogen minus that H+, okay? So this form of lidocaine exists then at pKa 7.9. So you know that at physiological pH, whether it's in our blood, you know, relatively saliva, they have very similar pHs. Only 25% of lidocaine is in neutral form, and 75% is in the conjugate base form. How do we know this? Again, additional separate measurements have already been explored, you know, well, have already been done to explore what this lidocaine, you know, what the actual forms of it are according to pH. So the active form right here might not be able to penetrate all the way through because of its strong polarity, right? This has a real strong polar group. I mean, it might be attracted to the head group here on that phosphobiolipid layer, but it can't tunnel all the way through. It can't pass all the way through. So it's going to have a little bit harder of a time. But what about the RN group? Remember, we talked about if you don't have this charge there, then maybe we can reduce some of that polarity. We don't start talking, or the type of intermolecular force dominance. It's not so much ion dipole now. Now it kind of gets reduced a little bit, maybe to some of that hydroxyl, hydrogen bonding type of character. And hydrogen bonding is less strong than ion dipole. That just might be enough to be able to tease that lidocaine through from outside to inside the cell. So the H+ then is able, you know, to be able to be recombined with that diffusible form once it's inside the cytoplasm, because H+ will exist. And it'll be able to recombine with this active form of lidocaine, RNH+, to be able to deliver that numbing effect. So pretty neat example, I think. And I think that this is, you know, something that's going to be, you know, of interest to you perhaps later in your studies. But the pH again is less than the pKa, right? So the protonated active version is favored, all right? But the diffusible form has this. So there's some processes going on, and actually this is, you know, in equilibrium, as you can see with these arrows, where you're going to have some of the RN plus H+ character outside that's going to be in equilibrium with the active form. And once inside the cell, you don't worry about that H+, because that's just going to hang out here. That's not going to be able to penetrate through that phosphobiolipid layer. You're going to actually only pass that RN form of the molecule right through the bilayer, and then it'll respond to the H+ inside the cytoplasm to reform that active molecule for the pain. All right, so buffers and blood. Let's also talk about bicarbonates, okay? The basics of a buffer solution, you probably have heard this before, are the following: near equal combination of weak acid and its conjugate base. Notice that it's not necessarily talking about a strong acid or strong base, okay? So buffers are generally going to be used pretty much in its entirety, really, with weak acids and weak bases. In our bodies, we have weak acids, weak bases in our bloodstream and in our saliva. Obviously, in our contained organs like the stomach, we have very strong acids, but that's self-contained. In that stomach environment, we don't have buffers. So you can't think that we have a buffer system in our stomach, but as we progress through the digestive tract and we go through the small intestine and large intestine, it is completely reasonable to expect that there's probably going to be some weak acids and or the conjugate base present, and that might help facilitate uptake of certain nutrients. Buffers resist changes in pH, okay, if small amounts of acid and bases are added, and they do not comprise strong acids and bases. In other words, I don't know if you guys have seen the movie Alien or Aliens. I mean, they made, you know, several of them, but the blood of the alien essentially is sulfuric acid, so which would be a strong acid, and that's why it burns whenever, you know, it would open up and it would splatter on whoever that was trying to get it. But that's the whole point, you know. Do you think then that the alien form, if it had sulfuric acid in blood, would actually have a buffer, a good buffering system? No, because it lacks the weak acids and the conjugate bases. So in our bodies, we have the bicarbonate buffer system, the H2CO3 and HCO3 minus, or you have here H2CO3 or NaHCO3. Usually this other thing, this other format here that I have in parentheses is given in laboratory experiments and so-called in vitro, you know, in the laboratory, in glassware, really try to, you know, outside the body, kind of mimicking the live clinical environment, laboratory studies. Okay, so let's talk a little bit about that. So water plus carbon dioxide, and this also corresponds to carbonated beverages, CO2 dissolved in water, whether it's your Perrier or whatever other kind of, you know, drink that you're enjoying. You know that if it's pressurized, it'll have the carbon dioxide in the water. This also applies to sodas, of course. But know that it rapidly forms with water to create carbonic acid and water. But also, because carbonic acid is a weak acid, guess what? It goes back the other way, and also water and carbon dioxide are also going to exist. So you have this rapid equilibration between both processes, water and carbon dioxide going to carbonic acid, and then, of course, you know, carbonic acid with water, you know, equilibrating to the left to form the carbon dioxide in the water. This is where temperature is going to really play a role. If we're going to keep things cool and like in the refrigerator, then the carbon dioxide is going to be able to stay in the water relatively easily. If you let that bottle warm up to room temperature, well, you're going to have some of that carbon dioxide going into that headspace between the liquid and the cap. If it's closed, if it's open, then you can forget about it. I mean, open up that bottled water, especially at room temperature, and the carbon dioxide will quickly leave, and then your water will go flat, which actually sounds kind of silly to think about it like that. So this is the form of carbonic acid, by the way. It has these two hydroxyl groups, okay, along with the carbonyl, so pretty interesting. And you can also see that this is a polyprotic acid. It would have two different Ka values, okay, because you have two hydroxyl groups. So one for each hydroxyl group. This also equilibrates, though, the carbonic acid in water to hydronium ion and the bicarbonate ion. Okay, so you have the carbonic acid is the given acid, and then its conjugate base is the bicarbonate base. With respect to water, well, that's acting as the base, and it goes to this conjugate acid, which is the hydronium ion, right? So there you have your structures, and you have the bicarbonate structure here too. I show you that the hydrogen is lost from one of the hydroxyl groups on that structure. Physiological pH is 7.4 in blood. The pKa1, and here's where we indicate, because it's a polyprotic acid, the pKa1, so on one of those hydrogens coming off that carbonic acid structure occurs at 6.1, okay? The second one, well, and that's in blood at 6.1. In water, it's about 6.35. So there are differences in having our blood versus water dissolution of carbonic acid. So you can see how that difference is. Now the pKa2, guess what? Every time you're trying to lose a second hydrogen ion, things get way more basic. So that's also something to keep in mind. Not that we're going to be quizzing you so much on all the different polyprotic acids and whatnot, but just know that you lose the first of the polyprotic acid, like carbonic acid, is 2 hydrogens to lose. The first one will always be more acidic than the second one, okay? And so here you have the pKa2 of carbonic acid being at 10.3. If you add phosphoric acid, which has 3, that would start at a low pH, which would mean acid, and it would gradually go all the way up to base, okay? It does, in other words, it's not going to go acid, base, acid in terms of these pKa values. So because physiological pH 7.4 is greater than pKa1, and we're talking about in the blood, okay, we're at 6.1, the conjugate base, HCO3 negative, is favored. Okay, this is why carbonic acid is not going to continue to exist intact as an acid in our blood if we drink it, or if we somehow get that directly into our veins, I guess. Now, let's talk about that. What happens if you interrupt that? So interruptions of homeostasis, meaning the regulation of the biological environment. If blood carbon dioxide level happens to increase, blood pH is going to decrease. What is this called? Respiratory acidosis. So this is where you breathe out way too much CO2 than your O2 inhalation. This would be an example of holding your breath. So if you were to hold your breath for 5 seconds and then open up, breathe in for 1 second, and then hold your breath again for 5 seconds, this is basically a form of hypoventilation, and you would actually create respiratory acidosis in your body, okay? So your body has water in it, right? Your blood does, and you have a lot of CO2 then because you're holding your breath. You're not able to allow it to escape properly. That means that your body, your blood in particular, if you do this for long enough, and it may not take that long, but you're going to actually increase the acidity of your blood, and you're not going to have that much carbonate of, you know, that conjugate base of the carbonic acid present. And so how are you going to counter all of a sudden that greater acidity in your blood? Well, if it gets to a point where, you know, and this isn't the case where you're messing around, this is if somebody really is hypoventilating, then you would administer bicarbonate solution, for instance, through an IV. IV, because that's going to go directly into your blood, and that's what you need to get, you know, less acidic, like immediately, because you don't want to have to start worrying about your body going too acidic or basic. Alternatively, if your blood CO2 decreases and your blood pH then will increase, okay? What happens here? CO2 exhalation is greater than your O2 inhalation. So hyperventilation is something that usually happens, right? Respiratory alkalosis, they call this. Breathing too quickly. And this is where they try to say, well, I'll tell you in a minute here, but you have, you know, water content in your blood has very little carbon dioxide because you can't regulate your breathing well enough. And then you have H3O plus. So the CO2 is low, guess what? H3O plus is going to be low, but then HCO3 is going to be larger. So this is what, you know, you're going to be battling. You don't have enough acid content in your blood to keep pace because your CO2 is so low, okay? So you're going to have more base. So what you need to do is you need to inhale your exhale, basically. So this is where the paper bag comes into play. So you can start to breathe in some of that CO2 that you have already expunged. Because what you're trying to do is just keep that CO2 then around for a little bit longer to be able to get you to relax, okay? Now that has to do with respiratory acidosis and alkalosis, but in the, you know, metabolism of certain molecules like fatty acids and stuff, you know, your blood pH can also drop. And in that particular case, then the excess acid in the blood becomes too acidic. Whoa, so what do? In your blood, if you have too much acid relative to carbonate, and it's not related to breathing, so it's not related to the CO2 exhalation or O2 inhalation, then one might have to administer bicarbonate solution. Again, you're going to do it through the IV because your blood is the issue. It's not, you know, you can't take a pill because that's going to go to your stomach, which is already decidedly acidic, and then it has to take time to actually process through your digestive tract. And so if you're having trouble with your body, you know, you're not going to be able to do that through the stomach. You can't do that through the GI tract. And again, if you have excess acidic elimination, meaning you're vomiting too much, then your blood pH is going to start to rise. So if you're having a massive loss of, and it could, you know, we say vomiting, but it can go both ends, right? You know, it can go out your exit, you know, you have a couple exit points, right? And if you're getting too much loss of this acidic material, then your blood is going to become too basic. And so what you might have to do is receive an ammonium chloride solution via IV. Again, this is just a weak acid. It's not something that's going to be terrible for your body, and it'll help get your blood back to where it needs to be. So these are just a couple of the different conditions. And your table 9.10 in your book kind of talks about it. It talks about you know, the metabolic acidosis and respiratory acidosis versus respiratory alkalosis and metabolic alkalosis. It's basically what I just explained to you, and you can review those because it's a pretty interesting story. Now, the very last part about this, I want to talk about weak acid action in the body, i.e. oxygen transport. So you guys know that red blood cells deliver oxygen from the lung, right? Well, what is this machinery, some of the machinery anyway, used to do this? So this goes back to chapter 8 where we were talking about kidneys. In this particular case, we're going to be talking about hemoglobin, okay? Because hemoglobin is some of the machinery that's used to do this. So you have 1, 2, 3, 4. You have 4 subunits, and each of those, you know, has a so-called heme group. The heme group consists of essential iron, you know, in the middle of what is and amounts to be a porphyrin ring. So all this weird architecture here all around that iron, okay, and they're all coordinated to it. You can see the red bonding environments, you know, going to that iron molecule, is the porphyrin ring. By the way, it's also known that the coronavirus has some kind of machinery that can be used to interrupt the bonding of the heme group to iron. And so this is one of the reasons why the coronavirus that we're dealing with now, that we have to be in quarantine for, is actually somewhat deadly to certain population groups, obviously, that are more susceptible to it by virtue of its impact with respect to respiration. So we have the other impact where it was going to mess around with some of the angiotensin proteins that are involved in blood regulation, but it can also deliver interruption with respect to the hemoglobin process. So that's another way that a virus can attack a body. So this is also kind of interesting from that perspective. You guys can reach out to me if you want, and I could provide you additional information if you really want to know more about that, about the coronavirus and its respiratory impact. But there's some interesting studies that continue to be done to try to explore, you know, what's going on with it. And it's actually one of the reasons why the anti-malarial drug chloroquine is being you know, considered to be something that could actually counter that, because that actually helps prevent the virus from attaching, you know, its protein machinery to the heme groups, and so it won't be able to interrupt that breakage of the heme iron kind of interrelationship. Anyway, we continue. So each heme group has an iron that can also bind to 2 more molecules. Well, guess what? One of those could be a histidine residue, okay, from the structure that's shown here. So you have this histidine, which is an amino acid, okay? So one of those is bonded to the iron group, and iron can have 1 more. Guess what it is? Oxygen. So how does it do it? It just has an oxygen just coming right off of the other end, okay? It's only bonded directly to only one of them. But what's really interesting is that the overall hemoglobin molecule with all its units and stuff, all the subunits, become, you know, spatially changed, whether you have oxygen attached to the iron molecule or not. So without, it's almost like iron kind of just sags with these other heme groups that are just like kind of just sitting there. As soon as oxygen is locked on, as it is in this oxygenated state, all of a sudden, you know, the orientation, then spatial arrangement then of the molecules that iron is attached to, all of a sudden they start to become a little bit more, I don't know, you know, symmetric, I guess, or maybe a bit more ordered. But, you know, histidine is still there. It's just in the presence of oxygen, those heme groups all of a sudden get uplifted a bit. And, well, at least in this orientation, how we're showing here. So each heme group, via the iron combined with one oxygen molecule. And each hemoglobin combined then to 4 oxygen molecules because there are 4 heme subunits in each hemoglobin, okay? So what happens here? So in a deoxygenated state, you see that you still have some histidines and stuff. And by the way, these heme groups and stuff, they're all still interacting with each other through amino acid interactions. Some of those are actually going to be hydrogen bonding. Some of those are going to be a little bit more serious, like these ion dipole interactions that would actually form a salt bridge. And so we had talked about that previously, and here it comes again. So in an oxygenated situation like what we're showing on the right, you don't have that salt bridge present. But in the if there is a salt bridge present, then as soon as that salt bridge forms, oxygen is released from iron, okay? So the formation of nearby amino acids that result in a salt bridge formation triggers the deoxygenation of oxygen, and that's done under acidic conditions. So you have the histidine residue. If an acid is present, then the amine group can become protonated. If the blood system, for instance, is a bit acidic, then that would give rise to the histidine all of a sudden taking on that charge. That amine becomes positive, for instance. Additionally, you also have the carbon dioxide being present in the acidic pH environment of the blood, and guess what? The carboxylate group then evolves as well. So that's where you get the positive from the amine, the protonated amine, and then you also have the carboxylate, and that the negative and the positives are then going to interact, form that salt bridge, release oxygen. Okay, so that's where we're at. That's where we're at here. So you have, again, what do you need in the acidic pH? You can have CO2 present and H+ present, or some kind of an acid. Guys, where do you think that might occur? What environment has that? Surely some tissues do. How about muscle cells? Like whenever you're exercising, and then people say, that there's a buildup of lactic acid, for instance, in your muscle, it becomes sore, right? Well, you have an acidic environment, lactic acid, and you also have carbon dioxide within that environment as well. So that would be an example of your muscles or some of your tissues having a favorable environment that might then trigger the release of oxygen if hemoglobin is going to be able to deliver oxygen to the body. So let's talk about it. Blood-rich CO2 is pumped from the heart, right, into the lungs through the pulmonary arteries. And here's a diagram, the cartoon that kind of shows all this. Arteries are blood vessels carrying blood away from the heart, by the way, so arteries away, and then veins are going to, okay? I don't know whether when I remember that, I'm sure you have your own ways, but arteries away and veins too. Veins are blood vessels carrying blood to the heart then, and in the diagram you have that color-coded scheme as well. In the lungs, CO2 in the blood is exchanged for oxygen. The oxygen-rich blood is transported then to the heart via the pulmonary veins, and then the oxygen-rich blood is then pumped from the heart to the many tissues and organs of the body through the systemic arteries, okay? In the tissues, the arteries narrow to tiny capillaries. Here, O2 in the blood is exchanged for CO2, and the capillaries widen into the systemic veins, which carry the CO2-rich blood, then back to the heart. So this is a closed system. You would consider this, our bodies are a complete closed system. Blood is coming from one area and ultimately gets recirculated. Well, during this recirculation process, you have an exchange, right? And what I'm trying to show you here is that you have CO2 and O2 being those exchange components, and your heart has, these individual chambers, and the chambers are definitely oriented in a particular way to be able to allow that to happen. So oxygenated hemoglobin is brighter red, and deoxygenated hemoglobin is a darker red. Here would be the chemical reaction that kind of shows this. You have hemoglobin attached to 2 oxygen molecules, and if this is all done in the bloodstream, right? So we're in aqueous conditions. In the presence of acid and carbon dioxide, you guys know that salt bridge is going to form, then there's going to be a release of oxygen, okay? Ultimately, the deoxygenated hemoglobin will then have this kind of character where you have an ionized form of the hemoglobin, plus, you have the carbon dioxide attached to the hemoglobin, no longer oxygen now, and it's also charged, right? And then you also have free oxygen then available. So let's take a look. Again, if you have localized pH, salt bridge forms and thus frees oxygen from the hemoglobin, okay? It releases it. This is especially the case for inside tissue. CO2 is exchanged for oxygen. Then, whenever there's a localized basic pH due to low CO2 content, so what happens is if CO2 is exchanged for oxygen, then hemoglobin now has the CO2 attached to it. Well, that leads to the so-called Bohr effect, and this is not Niels Bohr, this is a different Bohr, that has shown that in the localized pH environment, the effect in the oxygen-rich environment then results in this kind of uptake, where you have deoxygenated hemoglobin taking up, that proton and CO2 and taking it back to the lungs. So this is an acid-base reaction, really, and this is, you can kind of consider it a redox reaction too, if you want. I mean, here you have O2 on the right-hand side, the oxidation state of that is 0 because it's its ground state. Well, on the left-hand side, O2 in this hemoglobin molecule was -2, right? So, I mean, you had the loss there of the oxygen. You have a bit of reduction, if you will. And you can, look back and forth, just like, CO2 was one way and then H+ was another way. I mean, you can see how that can lead to the redox. And so again, showing the cartoon of the CO2 in blood exchanged for O2. So inside the lungs then, okay, so this hemoglobin's taken now the carbon dioxide back up to the lungs, right? O2 is exchanged, CO2 is then released, and this system continues ad infinitum until we expire, I guess, permanently. So there you go. That is the end of it. I hope that is enough of an information that we have going on. And with that, we conclude chapter 9, and then we will get ready for chapter 10. So thank you, and good luck with your homework and on the exam.